In this podcast, I bring the three top scientists who know more about fats and the brain than any others. Professor Michael Crawford, visiting professor at Imperial and head of the Institute of Brain Chemistry and Human Nutrition. Professor William Harris from the Fatty Acid Research Institute. And neuroscientist Dr. Simon Dyall, a director of the International Society for the Study of Fatty Acids and Lipids. To get inside our brains, I'm most interested in the points of disagreement, the contradictions, the evidence that doesn't fit the model. So often by stirring things up, we learn something new. Is it all about omega-3 DHA? So why is there evidence for EPA, sometimes even working better for some aspects of mental health, such as depression? What about phospholipids? Is it all about choline, which is just one of 5 phospholipids? Where does vitamin D, a fat-based hormone, fit in? These are some of the topics we'll be exploring. So, Michael, Simon, Bill, welcome to my podcast.
Thank you. Great to be here.
Thank you.
Thank you. Yeah.
So, Simon, so we have some understanding of how omega-3 fats or fatty acids actually build the brain. Can you give those listening a layman's guide to how the food we eat, rich in omega-3 and phospholipids, actually builds brain cells and connections?
Thank you, Patrick, and thank you for the invitation to talk today. So the brain is mostly fat. Around 60% of the dry weight of the brain is lipids. Of this, these are mainly in the form of phospholipids, which I think we're going to talk about later on. Phospholipids are made up of fatty acids, so fatty acids are attached to in the phospholipids. Around 8 to 10% of the fatty acids in these phospholipids in the brain are the omega-6 arachidonic acid, and around 12 to 15% of the fatty acids in these lipids are the omega-3 fatty acid, docosahexaenoic acid. So these two fatty acids, and omega-6 and omega-3, are very highly enriched in the brain. And Michael's seminal work in the 70s showed that this kind of enrichment is conserved across species. So there's a specific enrichment of the brain with these fats that we can get from the diet. The neurons are the brain cells that are involved in the fast communication. These are around 8% DHA and around 10 to 15% arachidonic acid. But it's not just how much is there, it's where it's there, it's the location. So neurons communicate via these things called synapses. They're kind of connections between the neurons via these chemical synapses. And DHA is very highly enriched at the synapse. It has a very, very important role in the communication between neurons. Part of the process of communicating between these nerve cells is via neurotransmitters, and DHA is released during this neurotransmitter activation. And it's been estimated that around 100% of our DHA is re-esterified and released and reconnected every day. And this composes quite high demands on the brain. So the DHA and the arachidonic acid that we get from our diet are found at very high levels in the brain, and they're found at very specific, important places within the brain. I was going to say
that those synapses, that's really where the talking is happening in the brain, across that gap, the synapse from neuron to neuron. That's where the communication is taking place, and DHA is bang in the center of that. Is that correct?
Exactly, yeah. So neurons aren't physically connected. They have this little gap between them. And what happens is there's a nerve impulse, a neurotransmitter is released, and it diffuses across to the next neuron. So it's how the neurons communicate. And the neuronal communication is basically how our brain works, how memories are formed, how we construct reality, is this communication. And DHA is very highly enriched at these specific locations within the brain. So it has a key role within communication between neurons, and it has a very, very conserved role across evolution.
And Michael, it was your seminal work back in the 70s. You looked at brains of many species, and you found in every single brain of any animal, it's that predominantly that DHA, the omega-3, and arachidonic acid, omega-6. Is that correct? And is that still correct?
Oh, well, we looked at 36 different species, Andrew Sinclair and I, and they were astonishingly all the same, bar little bits, but basically the profile was almost identical. Right across these 36 species, we've now expanded that to 42, and included marine species as well, which were again pretty well identical. Now, the interesting thing about that was that if you looked at the liver, which had a sort of more of a reflection on what the animal was eating, and its evolutionary pathway, that the liver was basically totally different by comparison with the brain. There were hardly any two species had the same kind of liver compositions, although there were kind of relationships. For example, herbivores versus carnivores were totally different. The carnivores having been slightly more enriched in EPA and DHA than the herbivores, which has in the liver very little DHA. But despite that, they still had the same DHA and arachidonic acid in the same profile, same proportions in the brain. The thing that's different was not the composition of the brain between species. What's different was the extent to which the brain has evolved, with obviously Homo sapiens at the top of the list, but not necessarily so much so. I mean, if you take the dolphin, for example, which is a very good example as to why the marine food web is so important. If you take the dolphin, it has 1.7 kilograms of brain. Now, the problem with dolphin, of course, is that it can only use half the brain at any given time, because it has to let one half sleep. If it doesn't do that, it'll drown. So it can't use the full weight of the brain. It has to sort of divide the responsibilities of what's going on into two different halves. So it's not quite the 1.7 kilograms of brains that's operating. But if you compare that, at any rate, even half of that, with a similar weighted land-based animal like a zebra or a horse or a lion even, which is a carnivore like the dolphin, then if the lion's only got about 340 grams of brain, it's much the same body weight as the dolphin, and the dolphin has 1.7 kilograms compared to 340. So I think that really clinches the fact that the marine food web was important to the evolution of the brain.
Now, and if we go backwards from docosahexaenoic acid, that's 6 double bonds. Before that is DPA, pentanoic acid, 5 double bonds. Then EPA, which I really want to talk about because it's EPA and DPA and DHA that we get most in fish oils. And then further up, and there's a couple of steps in the way from EPA up the chain, we get to alpha linolenic acid, which is the omega-3 that we find in plants and leaves. And I wanted to ask you, Bill, because there appears to be a lot of evidence for EPA, although not particularly present, I believe, in the synapses of the brain, being particularly beneficial for mood, perhaps more so than DHA. And also I noticed that your recent study using UK Biobank data found just as beneficial an effect from non-DHA omega-3 fatty acids as DHA fatty acids. Can you talk a bit about EPA and its effects on the brain, our mood, or anything related to mental health?
Sure. And again, thank you for the invitation to be on this panel. It's a lot of fun. I've already learned a ton from Michael just listening to him. I didn't know that half a dolphin brain sleeps while the other half works. I mean, that's amazing.
I wish I could do that.
Yeah, right. So EPA, yeah. So that's the 20 carbon omega-3 with 5 double bonds. And it is one of the 2 classic ones in fish oils. The DPA really isn't that much in fish oil, a little bit, but not much. DPA is a whole nother conundrum at the moment. But EPA and I guess you'd say mental health writ large. The idea that that EPA is maybe more helpful in depressive disorders has come from a large number of randomized trials, which, when looked at in aggregate, seem to show that it's those treatments that are richer in EPA than DHA, because you can have a higher amount of EPA or a higher amount of DHA in any given omega-3 product. The ones that are richer in EPA are the ones that seem to affect depressive symptoms better than the ones that are richer in DHA. Now, I don't know that means that the EPA is better. I don't think anybody's worked that out yet, what's going on, but the fact that EPA is not physically a part of the brain structure suggests that this is probably an intravascular effect inside the capillaries of the brain, still within the blood system, probably, but not actually in the brain itself. But I don't think we know, and I'm certainly open to hearing what Michael and Simon have to think about EPA and mental health.
Yeah, Simon, what's your take?
Thanks, Bill. Very interesting. Yeah, EPA. So we've done some work. We've done a systematic review looking at the different trials. The evidence at the moment does suggest that EPA at a greater than 60% of the overall supplement. So EPA to DHA, EPA is more than 60% of it. So high EPA does appear to be the more effective.
This is for depression, is it?
For depression, yeah. When you look at the brain, interestingly, Sophie Lay's done some great work looking at microglial cells. And microglia are the kind of resident immune cells within the brain. And they actually contain about 4% EPA. And the DHA is very, very low. So microglial cells actually contain a reasonable amount of EPA. So it is found within these immune type cells. And there is evidence that EPA is more effective when there's an inflammatory component to the depression. So depression is obviously a highly heterogeneous condition, but with treatment-resistant depression, there tends to be more inflammation, and EPA may be effective augmenting treatments or standalone in depression with an inflammatory component. So it may be related to their role in microglia, it may be related to EPA's role in resolving inflammation, anti-inflammatory effects. So there may be that kind of mechanistic view or approach to it. But the actual head-to-head trials between DHA and EPA haven't really been done. So it's based on fairly limited literature, but the literature we do have does support a more effective role with depression, major depressive disorder, for the high EPA supplements, possibly related to an inflammatory phenotype to the condition.
Do they have any effect on the serotonin receptors as such?
So there's some great early work looking at very low omega-3 diets on neurotransmitter levels, and there is clear evidence that rodents that are given very impoverished omega-3, so omega-3 deficient diets over generations, have alterations in serotonin transmission, alterations in serotonin and other neurotransmitters. So that there are interactions, but it's not clear exactly what they are.
Michael, any thoughts?
No, I think it's been well covered. That the likelihood that they could be involved in the astrocytes is important, as well as arachidonic acid could well be important in the astrocytes, but nobody's really looked at that. And I think I agree with Bill that there's almost certainly a vascular component that is important for this effect. But we need a lot more data and more precise data. One thing I will say, however, that is relevant to this question of everybody talks about fish oils, is that if one looks at the diets, we don't eat fish oils. The epidemiological evidence for if you like, for cardiovascular disease and major depression is related to fish consumption and fish and seafood consumption. And of course, if you look at the cod mussel, for argument's sake, cod mussel phospholipids contain 47% docosahexaenoic acid. I'll repeat that, 47% docosahexaenoic acid. And they have very little EPA or any of the other precursors, or for that matter, any other polyunsaturated fatty acids. The mussel itself is extremely rich, specifically in docosahexaenoic acid. And this makes a lot of sense from many different points of view, and particularly from the point of view of the fish, which has to accelerate to 30 miles an hour in about 0.3 of a second to catch its prey. Actually, when you eat fish or seafood, when you eat the meat, it's a very different story to the oil. The cod liver oil, for example, compared to the meat, the cod liver oil contains about, Bill will correct me if I'm wrong, about 12% EPA and about 8% DHA. And
Correct.
it's in a different form. It's a triglyceride rather than a phosphoglyceride, whereas the stuff in the cod muscle is, of course, practically all phosphoglyceride. So the epidemiological evidence is really quite different to what we're talking about when we talk about supplements and eating fish or seafood.
Well, and obviously eating fish is a very different thing than taking an omega-3 supplement. There's many nutrients in fish, and there's many nutrients in things that you're not eating because you are eating fish.
Yeah, that's right. Yes, you take the supplement and off you go to McDonald's. That's fine.
Yes, right.
And talking about supplements, what about algal DHA? Because not only are there some people who are rather plant-based and a bit averse to eating fish for whatever reason, but also we have this situation of a growing population of people and a shrinking population of fish. And if we can grow lots of algae, and I'd also quite like to know whether the algae actually contains that much DHA or whether it's processed in some way to concentrate it or produce it. But is algal DHA just as good as either fish oils? Probably not fish, because we've learned there's a lot more going on in there.
Yeah, well, algal DHA, and we're not talking about seaweed here. We're talking about a microalgae, which is a single-celled organism in the ocean that is the basis of the production of EPA and DHA. And the fish eat it. Fish don't make omega-3s any more readily than mammals do in general. So it's not that the fish are making omega-3; they're eating omega-3, which begins again at a single-celled plant in the ocean called microalgae. There's many species of them, and when that algae, that particular species, is grown in culture and in mass, and the lipids are extracted from that algae, it's the same chemical as we get in fish or fish oils. So EPA and a microalgae or DHA is the same stuff. And I just reviewed a paper comparing a microalgal EPA and DHA product to a fish oil product in terms of effects on blood omega-3 levels, and they're equivalent, so, which is not a surprise. They should be.
No.
So, yeah.
So they're not out there fishing for algae. They're actually growing algae effectively in tanks and then concentrating the DHA out of it. Is that how it works?
Yeah.
And because I often wonder, because these algal DHAs are remarkably low in EPA, and I didn't really understand why that was.
Well, certain species are, each species of, well, not each species, but there are species that are pretty much DHA makers, and there are species that are EPA makers.
Yeah.
And there are species that are both makers.
Mm-hmm.
So it's just, this is not genetic engineering, which could be done to produce a higher omega-3 in microalgae, but there are species existing in the ocean. It's just a matter of finding them and then culturing them that do both.
Can I?
Bartek originally produced DHA from algal sources to provide for infant formula a long time ago.
Right.
It was taken over by DSM, but it's basically done by algal incubation.
Simon, you had a point.
Yeah, I would just interject a little bit here. So Michael kind of alluded to it. So there are differences in the forms. So you can get capsules that contain what's called ethyl esters. You can get capsules from algae that contain phospholipid forms. So as Bill was saying, so in terms of having effects on blood cell levels, in terms of having effects on absorption, they're probably somewhat similar. There is some evidence that getting into the brain, phospholipid forms are more effective because they're specific lysophospholipid transporters to get across the blood-brain barrier. So there is potentially more benefit for a phospholipid form rather than an ethyl ester or a triacylglycerol form. So there are subtle differences in the form of the supplement that may have different effects on the brain.
So
Yeah, I think we need to make it clear that you can take an ethyl ester, an ethyl ester, in properly said, I guess, or a triglyceride capsule, and it will be ultimately processed into, some of it will end up in phospholipids in the body. Just because you're taking an ethyl ester omega-3 product doesn't mean it's an ethyl ester form in your blood or in your
Indeed,
tissues.
yeah. So the
Right.
liver's involved in processing and packaging,
Right.
shall we say? Yeah. So yeah, we're in agreement there. Yeah, definitely.
And you mentioned lysophospholipid. Do you want to explain what that is?
So essentially, a phospholipid, the main glycerophospholipids have a 3-carbon backbone. Position 1 tends to be a saturated fat. In the middle position tends to be an unsaturated or polyunsaturated fat. And position 3 is a phosphate. And attached to that is a different head group. And the different head group determines whether it's a phosphatidylcholine or a phosphatidylethanolalamine. And a lysophospholipid has just got one fatty acid rather than 2 fatty acids attached. And there's some really important work that was done that shows that there is a specific lysophospholipid transporter that's involved in getting lysophospholipid DHA into the brain. So there's 2 routes for getting omega-3s into the brain. One is via simple diffusion, and one is via this particular transporter of a slightly modified phospholipid, this lysophospholipid. I don't know whether I've lost your listeners there or not.
Well,
Patrick.
let me step back and explain a little bit here. And thinking of a capital E is very helpful, because these fats are made of a backbone of triglyceride. That's like the vertical part. And then we've got the 3 spokes of an E. And that's what we're talking about here, one of which is a fatty acid and the other which attaches a phospholipid. So what we've got really happening in the brain, as I understand it, is omega-3 in this context attached to a phospholipid. When I think of phospholipids, I think of eggs. I think of fish as the 2 richest sources. And let's talk about those phospholipids. They come in different forms. They're always phosphatidylcholine, phosphatidylserine. So there's always a phosphatidyl part to it. And I remember, Simon, you having a conversation with Professor David Smith, our homocysteine B vitamin methylation expert, and he was really talking about the fundamental role of phosphatidylcholine DHA, which is PCDHA. And he was making the case that in Alzheimer's, there are 2.5 times less PCDHA in the blood and 20% less gray matter. I believe he was saying, isn't this the really important thing? And you were saying it's not the only phospholipid. So tell us about these other phospholipids.
Thank you. Yeah, it was a very interesting conversation. Obviously, David's done some great work. We're looking at the homocysteine cycle and methylation and interconversion of these different phospholipids, without getting too complicated. So phospholipids, as you say, are like an E shape. At the top of the E, you tend to have a saturated fat. The middle of the E tends to be a polyunsaturated fat. And the bottom of the E is the head group that determines which particular phospholipid there is. So phosphatidylcholine is found at very high levels within the brain, tends to be on the outside of the membrane. Phosphatidylethanolalamine tends to be on the inside of the membrane, along with phosphatidylserine, that's on the inside of the membrane. Phosphatidylinositol tends to be found at very low levels. That's important for cell signaling. There are, without getting too complicated, there are what's called plasmalogens, which are slightly modified versions of this. But within the brain, DHA is found particularly in phosphatidylethanolamine, PE, and phosphatidylserine, PS. So they're the main ones that it's found in. So that's within the brain architecture. Circulating around in the blood, the phosphatidylcholine version, that's the one that when a fatty acid is removed is involved in the transport into the brain. So circulating phosphatidylcholine is very important because it's involved in getting DHA into the brain. But once it's actually in the brain, it tends to be more in the PE phospholipid. And David's work really nicely shows that the B vitamins and the homocysteine cycle are involved in converting the PC to the PE, which is very important to consider in the role of omega-3s in terms of nutrition and trials, and the role of B vitamins in nutrition and trials. And it's very important to consider the metabolic roles of these particular nutrients and the synergy and the interactions between them. So the omega-3s need the B vitamins in order to have this conversion of different phospholipids, and the B vitamins need the omega-3s in order to make the homocysteine cycle work. So there's this important interaction. But in terms of the relative importance, so Bill can kind of chime in here as well. So looking at changes in the brain with dementia is quite hard to do because of the post-mortem, etc. But there tends to be decreases in the omega-3s in key regions like the hippocampus and other stuff. But because they're most highly enriched in the PE and the PS fraction, that's why they're more kind of noticeable within the brain, having the changes. Whereas circulation wise, because the PC is involved in getting into the brain, that's why the circulation PC may be more important in that context.
So just for our language, we have phospholipids, and there are 4 main kinds: phosphatidylserine, PS, phosphatidyl'inositol, PI, phosphatidylethanolamine, PE, and phosphatidylcholine, PC. Just before turning to you, Bill, there's often been a general view we don't need to eat phospholipids because we can synthesize them in the body. But there was a very good article in the British Medical Journal called Choline in Crisis. So that's the PC. And also a study in which women in their 3rd trimester of pregnancy were given either 480 milligram of choline or almost double this, 930 milligram, very hard to eat. I mean, certainly impossible to eat on a plant-based diet. And the researchers then tested babies' information processing speed at 4, 7, 10, and 13 months. And not only were the babies of the mothers given the higher dose faster, but also the longer the mother had been given even the lower dose, the faster the child's reactions. So are these phospholipids, although semi-essential, really something that we need to eat more of? And obviously eating fish will give us phospholipids, but what do we need to eat to optimize phospholipids? That's my question.
Well, any meat product is loaded with phospholipids, but so I guess even land animal or sea.
And
eggs
are particular.
And eggs are good, yeah. But back to that study you just mentioned, they were giving them choline. They were not giving them phosphatidylcholine, right?
Yes, exactly.
So maybe there's a difference. And we do make, if we have enough choline, we can make choline. It's conditional, and from that we can make phosphatidylcholine in our livers. And whether we need more phosphatidylcholine versus more choline itself in the diet, I guess and I'm not an expert on choline, so I'll let you talk more about it. You know more about it than I do, but that's my thought. We need to differentiate between choline and phosphatidylcholine.
Now, I've heard it said that if a mother has an insufficient supply of either omega-3 or phospholipids, possibly choline, their brain size can shrink because the baby will rob, or the fetus will rob the mother of its essential fats to build its own. A case of mummy, I shrank your brain. Is this true?
I don't know that evidence. I can imagine it, but I don't know the evidence if it's true or not. What do you guys think?
I've never heard that one. I think the brain would be very resilient to changes, so there may be decreases in more circulating levels, but I wouldn't.
Yeah,
I'm not familiar with that. Yeah, I would say that there's been some really great work looking at DHA and effects on preventing preterm births. So that there's some really great work looking at. There's the Susan Carlson and Maria Mikides have done some really great work looking at the role of DHA in preventing premature birth and prolonging the gestational period. So it's very, very important, and a lot of mothers may be decreased in their fish intake because of worries about mercury and all these kinds of things. So DHA is phenomenally important during pregnancy, but I haven't heard about the effects actually detrimentally on brain size.
And the other side of that really was women reporting baby brain. So often I've heard I felt like I had half a brain. So something happens to some women during pregnancy that decreases their cognitive powers. Now, I know, Michael, you've done quite a lot of work on pregnancy and women and their fatty acid levels and the outcome in the babies and their neurodevelopment. Tell us a little bit about that.
Well, there's little doubt that high-risk women, and by that I mean women who are at high risk to producing a low birth weight baby or a preterm baby, that there is strong evidence that the brain is shrinking from magnetic resonance imaging. And the thoughts definitely are that this is robbing the brain by the fetus. For the placenta, you see, it steals arachidonic acid and docosahexaenoic acid. The proportions of arachidonic acid and docosahexaenoic acid in the fetal circulation are many times higher than they are in the mother. And all precursors, and we've published this, all the precursors, including EPA and the DPA, and all the precursors of arachidonic acid, linoleic acid, but not dihomogammalinic acid, or the precursors are all sent back to the mother. They're not, if you like, they're almost rejected by the fetus. So a lot of our work shows that if you look at pregnancy and follow the status of the mother during pregnancy, the arachidonic acid levels go down, and quite significantly. And in fact, the placental biomagnification of arachidonic acid is stronger than the biomagnification of docosahexaenoic acid. So there's a lot going on here which would suggest that the mother is being deprived of both arachidonic and DHA in favor of the fetus. Don't forget that the fetal brain development is going to reach 70% of the total adult number of brain cells by the time the baby is born. So there's a huge theft by the placenta stealing arachidonic acid and DHA. And it would not be surprising that this, in fact, is part of the explanation for the shrinking brain. It's not universal, not every woman. The brain shrinks, but the high-risk women, there's certainly significant evidence of that.
High-risk women and politicians, isn't that the problem we have?
That's another story.
That's another.
Another controversy that I'd like to sort of point out, but it kind of relates to this. When you talk about that phenomenal, production of neurons in the fetus, we start to realize the amount of energy that is going towards making DHA, and ultimately from the plant forms alpha-linolenic acid, which you'll get lots of in walnuts and pecans and chia seeds and flax seeds. And on the other side, the arachidonic acid, we have the first omega-6, in the seeds and the seed oils, sunflower, sesame, and so on. And the process of turning those into either arachidonic acid or DHA for the omega-3s, it's an expensive journey. It involves lots of enzymes and minerals like zinc and magnesium and vitamins and so on. So I just wanted to give that introduction to this whole controversy in the world of functional medicine, which at its most extreme says omega-6 is the demon, in the sense that it's this very high omega-6 to omega-3 ratio in our modern day diet from processed oils like soya, peanut, sunflower, corn. So too much omega-6 is kind of robbing our ability to produce omega-3, possibly a lack of these cofactor nutrients like zinc, magnesium, vitamin C, and so on. But here we're hearing that omega-6 arachidonic acid is absolutely vital. So what's this omega-6 versus omega-3?
I think they're probably talking about linoleic acid, because I think the diet is swamped with linoleic acid these days. And it's unnatural to have such high levels of linoleic acid. And it's very clear what the placenta does. It's making a clear choice. It's sending linoleic acid back to the mother. The fetus doesn't want linoleic acid. It wants arachidonic acid. And there's a very strong biomagnification of arachidonic acid. And the same applies to all the precursors of docosahexaenoic and oic acid. They're all sent back. And so effectively, the conversion by the fetus of either arachidonic acid or DHA is pretty well academic, because by the time you look at the fetus, and this is human data which we published in 1976 in the Lancet, if you look at the human data, the human fetus has fetal phospholipids both in the liver and the brain. There's very little of any of the precursors, therefore, to make any kind of bioconversion process viable. Nature doesn't want to have to build things. She wants the prefabricated units so she can slot it in. We've got to realize that the fetus is a very fast growing system. And it's not just the fetus that's growing, the blood volume's growing, the uterus is growing, the placenta is growing. So there's one hell of a lot of stuff growing during pregnancy that people forget about. And they all demand, in particular, the placenta is dominated. The placenta, all the phosphoglycerides on the placenta are dominated by arachidonic acid, with perhaps a little bit of a section of the ethanolamine phosphoglycerides, but DHA is still pretty minimal in all of the phosphoglycerides in the placenta. And if you look at the immune system, again, it's absolutely dominated by arachidonic acid by the time the infant is born. And then there's the vascular system. The endothelium is arachidonic acid rich, and so on. So it's and the muscle is arachidonic acid rich, and the red cells are arachidonic acid rich, and these are all expanding during pregnancy. So I think the idea that omega-6, if you just talk about omega-6, is unscientific. You've really got to think about these fatty acids in their own rights.
So does that
Amen.
mean I should eat less tahini? And what should I eat to get my arachidonic acid? And where do the whales get their arachidonic acid from? Because they've got bigger brains than all of us.
Yeah, the dolphin, funnily enough, if you look at the dolphin lipids, if you look at the liver lipids and the muscle lipids and the heart lipids of the dolphin, what you find again is both arachidonic acid and its elongation product, adrenic acid, is present, the two together, in a higher concentration in both liver and muscle than DHA. And there's very little EPA and not much DPA in the omega-3 series in either. When you get to the brain, of course, it's a different matter, because again, as Simon has already commented on, there's selective use of these fatty acids. That astrocytes are different to neurons, and neurons are different to synapses, and there are differences in different regions of the brain. It's not a simple issue, and I think it's quite wrong to be constantly going on about omega-6 and omega-3, when in fact each of the members of the two families have their own distinctive functions and operations when you look down at the biological detail.
What should we eat to get this arachidonic acid rich intake?
Eggs.
That's
a good answer. That's a good answer. Yeah, liver's rich.
How many years?
Wonderful, and meat is rich as well.
Yeah, meat, eggs, liver. How many years have we been told to not eat eggs because they will raise your cholesterol? What's your take on that?
Oh, are we going to go down that road?
Well, no,
we haven't got time for that.
One word. You can do it in one word if you like. Yeah.
Yeah, it was misguided.
Here.
No,
agreed.
I was looking at one study which looked at biomarkers for dementia, and the two strongest in this particular study was a raised homocysteine, e.g., lack of B12, folate, B6, and a low cholesterol. I know in America you have different figures, but here the unit was 4. If your cholesterol is below 4, which probably would only happen if you were prescribed statins unnecessarily, it actually increased the risk of dementia. And I believe, what is it, 25% of the body's cholesterol is in the brain? What does cholesterol do in the brain?
I thought we were talking about omega-3.
Well, fats was
Yeah,
our...
but cholesterol is actually very important. It's a very interesting
Yes, indeed.
issue, is cholesterol, because it doesn't matter what level of cholesterol you've got in your blood. The brain considers cholesterol so important that it makes its own. And of course, it's used in all cell membranes, and particularly in the myelin and all the rest of that stuff. But it's basically a major component of all cell membranes in the brain.
Now, talking of brain fats in a sort of broader context, because we've mentioned phospholipids, omega-3, omega-6, just touched on cholesterol. I'm happy to have any other comments about that. Vitamin D, for me, is a bit of a mystery, because we're seeing a lot of studies. People who have low vitamin D have something like 19 times greater risk of having cognitive decline. People who have high level of vitamin D have something like 4 times less risk of developing Alzheimer's. So the evidence in favor of vitamin D, which is a fat-based hormone for cognitive function, is very good. But the mechanism, that's what mystifies me. How is vitamin D functioning in relation to the brain? Has it got any connection with omegas, phospholipids, or anything that we're talking about? Simon.
Yeah, so vitamin D, yeah, as you've kind of alluded to, is maybe misnamed as a vitamin because it has hormonal roles. It's very important for gene expression. There's vitamin D receptors that are involved in regulating gene expression. And we know that omega-3s are also involved in gene expression potentially via retinoid receptors, and retinoid receptors, vitamin D receptors kind of work together. So there is an interaction between them in terms of gene expression, which regulates obviously cell fate, cell function. So they have multiple roles across the brain.
But sorry, Michael. Yes.
Well, David Andrews has reported some very interesting stuff about the significance of vitamin D3 in COVID infections. That's apparently the people who died from COVID infections were a very high proportion of them were deficient of vitamin D. But a point I would make, of course, is that in fact, if you eat fish and seafood, you'll get stacks of vitamin D as well. As well as vitamin A. Fish and seafood are not just simply omega-3. In fact, there have to be aspects of the marine food web which are containing arachidonic acid. Otherwise, where does the dolphin and the great whale get its arachidonic acid from? And as I said, the muscle and liver and the amount of muscle in the great whale, come on, I mean, there's over a ton of arachidonic acid in there because it's such a huge animal. And so the marine food web, actually, we don't know enough about these things. So people have been so interested in protein that nobody's bothered about the lipids and where we're getting them from in our food web. And there's almost certainly good sources of arachidonic acid in warm waters from the marine food web. And again, if you look at fish and seafood, it's not just the lipids that are important. There's all these trace elements. There's the copper, manganese, selenium, zinc. These are protective groups of the enzyme systems that the brain uses to protect its sensitive components from peroxidation. They live in the cell membrane, they live in the cytosol, they live in the subcellular components like the mitochondria and lysosomes. And don't forget that the brain is using some 20% of the energy in the adult, but it's only 2% of the body. And when the newborn baby arrives, it's something like between 60 and 70% of all the energy is going into the brain. So you've got to think that the cells themselves, the neurons and the synapses, using oxygen at the most phenomenal rate. It's quite extraordinary. It's much higher than were represented by that 20%, because there's quite a lot of the brain that actually doesn't use oxygen at all, such as the myelin sheath itself. So in effect, the brain is, for example, a requirer of antioxidant defenses, and it gets these using the trace elements. And they're the richest source of the trace elements are in marine food web, and particularly the sort of things like oysters, mussels, and crabs, and so on. These sort of the shellfish are especially rich in these trace elements. And I have a feeling that if we look at the iodine deficiency across the world, the global iodine deficiency is running at about, I think it's somewhere around about 2 billion people are deficient of iodine. And it's hardly surprising that they're all basically inland people. We did a study in Indonesia where 60% of the children had palpable goiter, but they were only children inland. When you went to the fishing villages, there was none of that. None of that. So, and we're seeing again iodine deficiency appearing in the UK. So the concept of fish just being omega-3 things is really false.
Now, my first teacher, as I was an undergraduate studying psychology and focused on schizophrenia, was a man called Dr. Carl Pfeiffer from Princeton. And he had a patient called Lisa, and the parents had learnt how to keep Lisa sane. She was diagnosed with schizophrenia, and that was to give her an oyster a day. They didn't know why it worked, but they knew it worked. And it was Carl Pfeiffer's studying of the oyster that led to zinc and really put zinc majorly on the map. I thought you'd like that story. Now, my simplistic view is that in the hot climate, we get omega-6, and in the cold climate, we get omega-3. And I often wondered if those migratory paths of whales and other such animals was actually in order to sort of reload with omega-6 in the hotter waters and return to the colder waters for their omega-3. Why do these?
That's what we've published on the wonderful migratory routes of the great whale, which goes up to the Bering Sea, and then it goes down to the huge migration route to the breeding lagoons, as they're called, in Bahia off the coast of Mexico.
So are they really eating? Are they getting more arachidonic acid in the warm waters?
Yes, of course.
From microalgae.
That's what they're doing.
They're
getting. They go up in the summertime to the Bering Sea, where it's permanent sunshine, and the waters are just teeming with food for them, and they get really hot on the food, and then they come all the way back down to the hive to do the breeding in the lagoons where they're getting their arachidonic acid. We've published on that.
Fascinating, isn't it? Now, we've spoken a lot about nutrients, but we really haven't touched on the other side, which are anti-nutrients. So I just wanted to ask about the effects of things like sugar and alcohol. Do the high sugar diet that is now the hallmark of the standard American diet, or the SAD diet, I think we call it, do they interfere with how we make brain cells and connections? And what about alcohol, which of course is a major antioxidant? What is it in our modern day diet, other than not eating the good stuff and lifestyle, that messes up fatty acid metabolism?
Well, alcohol has been shown to deplete the brain of DHA, so that's a definite factor there, is that a high alcohol intake does deplete the brain of omega-3s. So I think Joe Hibblin's done work on that, exploring that.
And what about sugar? What's happening here in relation to messed up sugar metabolism, insulin resistance? Is this impacting on fatty acids, or is it purely a fuel supply issue for the brain?
Apart from anything else, it dilutes the nutrient density.
And then there's a
Has
big
a global effect.
There's a big move towards plant-based diets and veganism. What impact does this have on the brain? And as we move forward in recommending a way to go, which obviously is towards a more whole food, wild food diet, what are your takes on that? And for people who are plant-based plant-based diets, what are your views? What would you like to say? Can they mitigate the effects, or is it just not the right way to eat?
Well, Michael has eloquently mentioned that we can't consider omega-3s and omega-6s as single things. We have to look at the individual fatty acids, along the pathways, and the high linoleic acid intake is having affects the omega-3 pathway, affects omega-3 incorporation. So the Western diet, seed oils, corn oils, these kind of things with very high linoleic acid, is detrimental to not only the efficiency of conversion from the short chain alpha linolenic to DHA, but also competition for incorporation. The ISFAL kind of position statement is that there's only two ways to increase DHA tissue levels. You either take preformed DHA, or you decrease your linoleic acid intake to help the omega-3 pathway. So a high seed, a high corn oil diet that's got very high linoleic acid is going to be detrimental to the omega-3 pathway. So we need to be thinking about ways of decreasing our linoleic acid intake as a first important approach.
What does that mean?
Patrick, so you wanted some controversy. I'll throw a little bit in here. I think we're a little bit in danger of seeing the world have an omega-3 centric view of the world, that whatever lowers omega-3 is bad, whatever raises it is good, no matter what. And I think that's not true. We've been looking at linoleic acid levels in blood as a marker of linoleic intake, of course, because you can't synthesize it. It can only come from the diet. And wherever we look, we see that the higher levels of linoleic acid are associated with better cardiovascular, better metabolic, meaning diabetes, health outcomes. We haven't looked at cognitive outcomes yet with omega-6 or linoleic, but it's entirely possible that a linoleic acid, even though it might lower levels of the omega-3s, is in itself a good thing, and it is producing a variety of molecules, of mediators that are healthy, and that lowering the linoleic acid intake may actually be counterproductive in terms of the major diseases, cardiovascular disease and diabetes. So I think we got controversy about, again, we shouldn't, like Michael said, we got to be specific about what fatty acid we're talking about. And now we're talking about linoleic, and I don't think it's a bad player. I think it's a good thing.
Well, one of my issues is that when we look at graphs of the ratio between omega-6 and omega-3 going up a lot, and talking about corn oil and so on, it seems that we're talking effectively about processed foods. And
we are, but I think you got to not look at omega-6 and omega-3 ratios for the exact reason Michael mentioned. You cannot pool these fatty acids together and presume that all the omega-3s have the same effect and all the omega-6s have the same. That's not true. And so we should get away from that ratio, I think.
Yeah, okay. But even so,
agreed.
Okay, so we all agreed on that. But even so, the issue is, if linoleic intake has gone up so much from processed vegetable oils, are they surely not principally in processed foods? And couldn't we, couldn't part of the problem just very simply be all these processed and ultra processed foods?
True.
Quite different.
That doesn't mean it's the omega-6 that's the problem.
Exactly.
Yeah, I think, Patrick, you asked really basically what's wrong with what we were eating, and I think this conversation of specifically focusing on one thing or another is not the way to go. For Bill, I think, and Simon, both agree with that, I'm sure. What you need to think about is, where did we come from? And we came from a chimpanzee which had a 340 weight brain, or 340 cc cranial capacity. Choose which you like. Now, the epigenetic expansion of cranial capacity from 340 cc's to between 1,500 and 1,700, between 28,000 and 32,000 years ago. The epigenetic force that did that could only have been food. It had to be the content of what was in food. So you need to think about what powered the encephalization of the brain. And what powers the encephalization of the brain is, first of all, you have to think is, what is the brain made of? And it's made of arachidonic acid and docosahexaenoic acid. So at its very simplest, you have to realize that the brain evolved in the sea 500 million years ago, when it's evident from all the studies that have been done on fish, the cephalopods, the amphibia, the reptiles, the birds, the mammals, and ourselves, all have this conservation of docosahexaenoic acid used in the visual system, and which started it all, and in the signaling systems of the brain. So for a start, a section of marine food web in the food structure is sine qua non. Now, to get arachidonic acid, that mainly comes from the land-based food web. So what we're talking about is that the power, the biological power that forced encephalization. Because don't forget, if you look at all land-based mammals, that as their body sizes grew bigger and bigger, their brain sizes grew smaller and smaller in relation to the body size. You start off with things like a marmoset or a squirrel or hyrax or something of that sort, or even a humble rabbit. And the proportion of brain to the body is over 2%. When you go up to the big animals, you go up to a rhinoceros, and it's 0.01%. What has happened is they got all the protein they need from the simplest foods, grass, leaves, and so on and so forth, but they can't make brains. But if you go to the marine food web, you see where we have these huge brains, many times our size. So it is sine qua non that the marine food web was a major component in encephalization. And so was the land-based food web. We have the best of both worlds. Now, when you think of that, it's all wild food. Just think of that compared with what you're talking about that's in our supermarkets today. We've got to start thinking about the need for the brain. What is the basis of all agricultural policies is protein. Protein is counterproductive. Rhinoceros gets all the protein it needs from simple foods, namely grass. It's only got 0.01% of the body size is brain. We have to think about how we can get, not back to, but how we can manipulate the food web so that we can start to get something that approaches the wild food web that can increase encephalization to a large size we had between 28,000 and 32,000 years ago, because the peak was 1,700 cc's. The brain capacity to cranial capacity, the most recent measurement is only 1,336 cc's, and we've seen this increase in mental health, which keeps on going up and up, and if a decline in IQ keeps on going down and down, then Homo sapiens is finished.
And on that
poetic and tragic note. I think it's a...
Oh, it's not tragic. It's actually a very positive note, because there's an answer to this problem. That all we've got to do is to start thinking about lipids needed for the brain and start developing agricultural policies that provide that. And that means starting to agriculturalize the oceans, the simple thing.
Yeah, create the conditions of existence that allow the marine food web to return to all our beautiful coasts, of which we have hundreds, if not thousands of miles in Great Britain as an example. Yeah. So we have run out.
The world is 71% water. The planet is 71% water. Of the rest of it, only a third is useful for arable agriculture. And we, according to foresight recommendations to government, there's really not much more land that can be brought into agricultural use. And in fact, the proportion of agricultural use is falling because of urbanization.
Michael, tell us what they've been doing in Japan on the basis of listening seriously to your recommendations.
Well, they've been doing exactly that. They've been farming the seed. You know, we have what do you call it? You have grass pastures for sheep and cows. The Japanese have grass pastures in the seed, seagrass, very simple. And they have kelp. Kelp, not only does it provide food, it provides iodine in particular and other trace elements. It provides fertilizers for land, and it does the same as the Amazon forest. It soaks up CO2. And they plant artificial reefs, which make use of the wealth of the marine food system, and things grow on it. Marine flora grow on these things, and that multiplies the productivity of the oceans around these two islands that Takahiro Tanaka has developed, and it's just phenomenal. In a few years' time, they simply tripled the productivity of the oceans.
So as we come to the end of our podcast, Simon, Bill, do you have anything to say? I know, Bill, I think you started your career here chasing salmon one way or other. Is that right?
Well, I was told to, and I did it. Bill Connor was my mentor, and he put me up to that, and it's been a great ride.
So any final comments before we close?
Let's do it again.
Yeah, it's been really good fun. Thank you. It's been always a pleasure to listen to Michael, and great to discuss things with Bill. It's been a real pleasure. Enjoyed it.
And in a couple of months, Michael, I think we're going to be celebrating your birthday. How old are you going to be?
Who are you talking to?
You, Michael.
Me? Yeah. What about it?
How old are you going to be at your next birthday coming soon?
94. 94.
And still as sharp as a razor.
I don't know about that. It's getting a bit blunter these days.
Well, you're still doing your own plumbing.
Yeah.
Very good. So, gentlemen, thank you so much for all your research over the years, all your wisdom and shedding light. This really was the Brain Fat Think Tank. Professor Bill Harris, Professor Michael Crawford, Dr. Simon Dyall, thank you immensely for your time, your expertise, your wisdom, and sharing it with us.
You're welcome. Thank you.
Bye-bye.